One of the ways I look at changes in specific protein or gene expression is through something called in situ hybridization. This gives me an idea of activity and of how much protein is being made in a specific brain region. For those of you vaguely remembering high school biology, remember that DNA makes RNA and that makes the protein. DNA is the blueprint for everything in our bodies, namely proteins, from cell membranes to neurotransmitters to receptors. At the minute, I'm looking for a specific receptor called a GABA receptor. These are found on cells, and changes to GABA receptors or GABA receptor expression will give me an idea of the physiological mechanisms at play in prenatal stress. GABA receptors are a complex set of receptors that are essentially Gandalf to the electrical currents in your brain. You shall not pass! That's called inhibition. The more GABA receptors are present and switched on, the less likely it is that the Balrog will pass to the next cell. In this case, the Balrog is an electrical current passing between synapses to convey information in the brain. So we call this the cell is inhibited. In general, inhibition is a really great thing and it keeps our bodies and our brains in check. I'm interested in whether prenatal stress changes levels of inhibition through GABA receptors, especially in the brain areas that are associated with stress and anxiety. So, a quick recap. If you watched the last video, you saw me taking rat brains and cutting them into tiny thin slices and mounting those slices on microscope slides. These were brains from prenatally stressed and control rats that then either received a week of chronic stress or were left alone. Then I tested them for anxiety and then took their brains so that I could look for changes in GABA receptors using in situ hybridization. Because this is quite a technical video, I'll split it into 3 parts. Part 1 is making the probes for GABA receptors. Part 2 is using the probes on the brains. And part 3 is the finally exciting part, that's getting the results. Step 1 of an in situ hybridization is making probes that will only bind to GABA receptors. The probe is a sequence of nucleotides, like the ones you would find in DNA or RNA. That would be A, T, C, and G for DNA, and A, U, C, and G for RNA. These sequences of nucleotides are a perfect complementary match to the sequence of nucleotides on the gene that I'm interested in looking at. Because it's 2019, I can go online and order the exact sequence I need for my probes. Once it arrives, I need to add a radioactive label to this sequence. This will be important later so I can visualize where the probes have actually bound, so I can then see where and how much of my gene of interest is there. I use radioactive sulfur, S35, which is bound to an adenine nucleotide. I set up a reaction in which an enzyme basically just adds this extra radioactively bound nucleotide to the sequence of probe that I have. After cleanup to get rid of all the extra stuff in the mixture, I am left with probes that have this extra adenine with the S35 attached to it.